Welcome, everyone. Thank you for coming. So today we'll be talking about Bayesian Optimal Phase II design, abbreviated BOP2. So I just had you fill out a survey. So let's talk through the answers. Oh, got two people filling it out. Okay, great. Let's look at the results. So first, what is the purpose of a phase 2 trial? Everyone, all two people said, determine whether the drug has sufficient activity to warrant further study. Fantastic. This is the correct answer. So we're all on the same page. Which of the following does the statistician need to select before using Simon's two-stage design? Select all that apply. The desired type 1 error rate, desired power, response probability of the drug under the null hypothesis, and response probability of the drug under the alternative hypothesis. That is great. That is correct. So with Simon's two-stage design, what it's going to give us is the interim sample size and the final sample size. So we don't provide that. And maybe I should just say, for the BOP2 design, it's going to be different. We will still provide the desired type 1 error rate, but with the BOP2 design, it's going to maximize power. So that's maybe the biggest difference between Simon's two-stage and the BOP2 design. With Simon's two-stage design, you're minimizing the expected sample size or the maximum sample size, depending on which option you choose. But with BOP2, we are maximizing power, which means that we have to decide for ourselves what we want the interim sample sizes to be and the final sample size as well. And we'll get into the details of that in a moment. Don't worry, that's not the only time I'm going to cover that today. But this is just a bit of a review, and I'm highlighting comparisons between Simon's two-stage and BOP2. So with Simon's two-stage, it's going to give us the interim sample size and final sample size. We will have to provide that for BOP2. What is the goal of Simon's two-stage design? I just mentioned this. We're minimizing the sample size, either the expected sample size or the maximum sample size. And again, with BOP2, that's not the case. With the goal of BOP2 is to maximize the power. Oh, we have some disagreement here. When using Simon's two-stage design, we stop the trial after the first stage if. So we have two people saying we are convinced the treatment is futile, and one person saying we are convinced the treatment is effective. So I am not someone who has conducted a ton of Simon's two-stage designs, and so this is not a perfect question. You may have used this design to stop early for efficacy, but I was basically going off the original paper in which you're stopping early for futility or for toxicity. So not necessarily, you would know better than I would how this is used in practice. And then match each term to its definition. So the prior, it's hard to read these, so I will go to. It's easier to read all the questions. Yeah. Okay, match each term to its definition. So the prior is the distribution of the parameter before any data are collected. The likelihood is the distribution of the data conditional on the parameter. And the posterior is the distribution of the parameter conditional on the data. So BOP2, it's a Bayesian design, and it's going to require more knowledge and use of Bayesian statistics than, for example, the Bowen design. With Bowen design, again, you're the experts, but based on my understanding of Bowen, you can sort of do it without really needing to know a lot of Bayesian statistics. With BOP2, it's going to be important to have an understanding of at least these three concepts. So we're not going to go into detail. I actually elected not to have any slides displaying Bayes' rule or Bayes' theorem. We're not going to talk about theorems, but it's important that you understand these concepts. So just to go through them again, the prior, that's the distribution of the parameter before any data are collected. So with Bayesian statistics, we're assigning a distribution to the parameter. With frequentist statistics, we assume the parameter is fixed. We don't know what it is, but it's just some fixed value. With Bayesian statistics, we're using a distribution to quantify our belief about where the parameter is. So we need to do that before we collect data, and then we update that distribution after the data are collected. And then we're going to make decisions based on the posterior distribution, which is going to incorporate both the prior information and then also the data that we observe. Are there any questions about these three concepts? Okay, cool. I think that was the last question. All right, any questions about these questions on this survey before I proceed? Okay, cool. Thank you for humoring me. All right, so before we dive deeper, I'm going to give you an idea of the structure of today's training. So with BOP2, there are multiple possible endpoints that you can use it for. So what I want to do is go through one endpoint in depth, specifically binary efficacy. That's the first option if you use the Shiny app. So we'll go through that in depth. I'm not sure exactly how long that will take, but we'll do that, pause for questions, and also take a break. And then we'll proceed after with the other endpoints, really just highlighting what makes them unique. So there's binary toxicity. You can monitor efficacy and toxicity simultaneously. You can have multiple binary efficacy endpoints. There's an ordinal efficacy possibility, and then finally, time to event. So yeah, this will be the structure of today's training. I have two assumptions and one request. So I actually know that already one of these assumptions is just incorrect, but that's okay. So I'm going to assume that you're a statistician. I already know that we have people here who are not statisticians. That's fine. I'm also going to assume that you have real-world experience with clinical trials, more than I do. I would say that I'm coming to this more as like a theorist. So I'm going to rely on your real-world expertise. My hope is that you'll critically evaluate what I tell you today in light of your real-world experience. So pretend that I'm trying to sell you a used car. You need to kick the tires before you buy. So this is my hope and my request, that you're not going to just take what I say as a given. Okay, so let's dive into our first endpoint. So the outcome we're going to assume is binary and good. I'm going to use the word response just to refer to this binary outcome. So the parameter of interest is P, which is the probability of a response. So this is what we're focusing on, P, probability of response. There are certain user-defined inputs that you will have to provide. So you will have to provide the interim and final sample sizes, the desired type 1 error rate, the null hypothesis response rate, alternative hypothesis response rate, and a prior within certain constraints. And I will tell you all about those constraints in a moment. But before we get into those, before we talk about the prior at length, you can visit the Shiny app on your own. So here's a QR code. I'll give you a second to scan it if you want to visit this app. You can also just Google, MD Anderson BOP2, and you'll get to the same result, I'm sure. You can also go to trialdesign.org, and that will take you to the same place. But the place you want to end up. All right, so MD Anderson, in their wisdom, has created this super useful Shiny app. If you've ever done a BOP2 design, or excuse me, a Bowen design, I'm sure you've used a similar app. So this is going to make our lives super easy. And you'll see here are the possible endpoints. That's the first thing we can select. We're going to talk about binary efficacy first. And then after choosing our endpoint, we select our interims. So here they have a default of 10, 20, 35, and 50. I don't know if that's ideal for your particular scenario, but this is what the default is. You can absolutely change it. But you can select your sample sizes when interim analyses are to be performed, separated by a space, and then the last number has to be the total sample size. And here they have 3 interims and then 1 final. So obviously that's another difference from the Simon's two-stage design where you just have 1 interim and then a final. With Bayesian statistics, there isn't really a penalty in terms of the statistics for having a bunch of interim analyses. You're just updating your posterior based on the data you observe. And it's not like with frequency statistics where you sort of have to like spend your alpha, and by looking at the data multiple times, you're inflating your family-wise error rate. So that's why you have so many interim looks here, that there isn't a statistical cost, you could argue. Now, there might be a logistical cost, depending upon how difficult it is to look at the data and do the analysis, or like wait and determine is something a response or not. So there are for sure logistical reasons for minimizing the number of interim analyses, but in terms of the statistics, not so much. And then we're going to have to provide our null hypothesis response rate, alternative hypothesis response rate, desired type 1 error rate, and then the prior. And it does have a like default that they recommend. Let's talk about the prior and depth. So we're going to assume that our parameter P is beta distributed. The beta distribution has 2 parameters. You don't have to have memorized what the beta looks like. I'm going to show you a bunch of images. And you don't have to know the formula. We're not going to talk about the formula. So let's talk about this prior. What does it actually look like? Here's a beta 1,1. It's just a uniform. So the uniform is a version of the beta. Here's the beta 0.5, 0.5. You can see, so the x-axis is running from 0 to 1. Obviously, our P as a probability has to be between 0 and 1. On the y-axis, we have the PDF, the density function. Here's a beta 2,2. Beta 2,3. So if the parameters are not identical to each other, then we get asymmetry. And then beta 3,2, asymmetry in the other direction. So just to give you some idea of different versions of the beta. How do we pick a prior within this constraint? It has to be beta distributed. So how do we pick? This is a good question. I'd like you to take a moment to write down what you think makes a good prior. So I'll wait. I'll give you, I don't know, 30 seconds to a minute. What do you think, before I've told you any other information, what do you think makes a good prior? You want to share?
What I know, it should be like broader than the posterior, right? And so more general than what you would expect. So you technically, once you add the data, kind of shrink the distribution towards the real values, right?
Yeah, I think that's a great point. That you might want sort of more variability in your prior, or have it be like squatter and wider. Because if you're going into this trial, this phase 2 trial, you could argue that you don't have a lot of information going into it. So you don't want to be like, let's have this very peaked prior over a certain value of P, because maybe then you're like gaming the system. Sure, I think that's a great point. Any other thoughts? You want to say more?
I guess like, I don't know, like this is very theoretical, but conjugacy property should also be desired, right? Like,
Conjugacy?
Yeah, like if you, I don't know, like if you really use that here, but.
Well, yeah, we are for sure using it. I just didn't mention it. So the beta distribution, when used as a prior for the probability of success in a binomial or Bernoulli, it is the conjugate prior. So we're requiring people to use the conjugate prior. It's not even an option to choose something else. Yeah. So we are going to return to this question, but I'm going to give you more information, and we can see if that changes your ideas about what makes a good prior. As on the way to give you more information about priors, we actually do have to talk about the posterior. So let's talk about like a single interim analysis. We're going to say little n is our interim sample size. We're going to say y sub n is the number of responses that we observe. Then that means that the posterior takes this distribution. So our parameter P, conditional on the number of responses that we observe, it's still beta, so it's a conjugate prior. Excuse me. But we've changed the parameters. So we've added our y sub n to the first parameter. And then we've added essentially the complement n minus y sub n to the second parameter. You don't need to know immediately like what that means. This is very abstract. So I can show you an image to try and convey what this implies. So here's a beta distribution with parameters 10 and 10. So I've increased the parameters by quite a bit relative to the images I was showing you previously. And you can see that we have this more peaked distribution. So as John Franco was pointing out, like when you increase the values of those parameters, we're decreasing the variance and increasing the height of the peak of our density. So when we add this y sub n and the n minus y sub n, we're going to get a more peaked distribution. So that's going to imply more certainty, which makes sense. As we collect more data, we're going to have more confidence, essentially. So I do need to give you a vocabulary term that's going to come up a bunch today, which is effective sample size. But first, I'm going to tell you about the mean. So the mean of a beta distribution with parameters A1 and A2 is A1 over A1 plus A2. The sum of the prior's parameters was A1 plus A2. The sum of the posterior's parameters is A1 plus A2 plus N. So by observing N observations, we increased the sum of the parameters by N units. And so this is why we call the sum of the parameters the effective sample size. So this is going to come up a lot. So the way that it's used is when we're talking about the prior, you could argue that we want the effective sample size to be low because we haven't actually collected any data yet. So we would want the sum of the parameters to be small. That's one way to think about what a good prior is. And then we'll collect data, that will be our N, and that will increase the sum of the parameters, and we're increasing our effective sample size. Like I said, this is going to come up again, so I'm going to pause here. Are there any questions at all about this concept of effective sample size? Okay, stop me. I should have said this earlier, but please stop me if anyone has any questions. All right, so let's talk about the default. In the Shiny app, the default is to set the mean equal to P naught, your null hypothesis value of P, and to set the effective sample size equal to 1. You can change this, but this is just the default. Another way of phrasing this is that the default prior is beta with parameter values P naught, or these are actually hyperparameters. The parameters of the prior are called hyperparameters. So the hyperparameters are P naught and 1 minus P naught by default. But again, you can change it. I'm just going to go back to the app so you can see it. Unfortunately, every time I move my mouse, the tabs are blocked. So I apologize. It's going to take a second. Okay. So use default vig prior with prior effective sample size equal to 1. So it's saying it's going to set the sum of the parameter values equal to 1. You can change it by choosing a different probability of efficacy and a different prior effective sample size. Okay. We have other options. I talked about a uniform prior that's going to make all values of P equally likely. That's beta 1, 1. There's a Jeffreys prior that's beta 1 half, 1 half. I'm not going to get into the math of what a Jeffreys prior is, you should have received an email from Erica with a link to some notes that I made that are on GitHub. I go into detail about what a Jeffreys prior is there, and I sort of demonstrate why this is the Jeffreys prior for the beta. But I'm not going to do that today. So you can definitely check out those notes if you're curious. But these are just other options for the prior. So I want to actually test this out with some real data. So if you came in person, I put you to work. I had you collect some data. So what I want now is a volunteer from the Zoom crowd who's going to provide some information. So I'll run through the information first. As I'm doing that, you can think if you want to be the volunteer, people on Zoom. So I want you to give me a real disease, a realistic null response rate, so null hypothesis response rate, a realistic alternative response rate, a realistic type 1 error rate, and a prior. So knowing now what I'm asking for, is there a volunteer on Zoom who's willing, is there someone on Zoom who's willing to volunteer for this? You'll have to speak up because I can't actually see anyone's images on my laptop.
Just remind people to.
Yeah, please unmute. All right, if no one on Zoom is willing to do it, is there anyone in the room who's
I
willing
can
to?
give you one. I
Thank
was
you.
I was
Thank you.
waiting for people to I was waiting for other people to speak up, but since people seem to be a bit nervous, I'll do it.
Thank you.
Okay. A real disease, gynecologic cancers.
Okay.
I'm literally designing assignments to stage with this right now. So let's see how the BOP2 does. So we're doing ovarian cancer. Sorry, can we go back to the list? So
Yeah.
type 1 error, I usually do 10% type 1 error for phase 2.
Okay.
The null is either 5 or 10%. So alternative is around 30 to 35.
All right, say the null again.
You can 5%.
Okay.
Then the alternative would be around 30%.
Okay. Then what about a prior?
We have no information. This drug, we have it in like other settings, but this drug has never been used in ovarian cancer before. So we don't know.
Okay. So do you want to use like a uniform prior then?
Yep. With like effective sample size of 1 is fine. The default would be fine. Yeah.
Okay. You said, yes, effective sample size equals 1. Then we're going to use a mean of the null value as well. Okay. So I'm going to make a plot. But I asked you for some things that didn't end up going into this function for the plot. And so I think it was, what was your alternative response rate?
I would say for a null of 0.05, I would put an alternative of 0.3.
0.3. Okay, great. And then type 1 error.
10%.
10%. Okay. Okay, great. Thank you so much. Okay, so we're going to use this prior, and then I collected data from some people in the room. So maybe we can start, who should I ask? Maybe, I'm sorry, I forgot your name. What's your name again?
Oh, yeah.
Yeah.
Yes.
So I had you collect some data. What was your N for the data that you collected?
10,
10. And how many successes did you see?
2.
2. All right, so now let's see if this works. Okay, so we have, it's a little hard to see, but the dashed line is the prior. So effective sample size of 1, and then we put the mean at 0.05. So it's like very right skewed because the null hypothesis value is so small. So the dashed line is the prior. The solid line is our posterior based on the data that Biao collected. And then what I've done is I've shaded under the posterior the area to the right of the null hypothesis value, which again was 0.05. So if you look at the top of the image, we have this probability. The probability that our P, the probability of a response, is greater than our null value, conditional on the data, is equal to 0.934. So it seems like based on our data, there's a very high probability that the probability of response is larger than our null. Are there any questions about that? Yeah,
Where does the alternative show up in this calculation?
it does not. Yes, so that is a good point. We're going to use the alternative. We're going to use the alternative hypothesis and also the type 1 error rate and all that jazz when we're figuring out the stopping boundaries. But before we get to the stopping boundaries, I just wanted to sort of zoom in on the prior and the posterior. But we will get to that, yes. Was there another question? Yeah. Yeah. I mean, I'm saying that, but we do have to sort of quantify what's high and what's low in the trial. And so we're going to do that in a moment. Yeah.
I have a question about
Yeah.
the prior.
Sure.
You said that it's the sum of the beta, the effective sample size is sum of the beta, right?
Yeah.
And if the beta gave an effective sample size of 1.
Yeah.
So doesn't it, isn't that the Jeffreys prior then, half plus half?
Well, the Jeffreys prior also has an effective sample size of 1,
Yeah.
but it has the equal, the parameters are equal to each other. I put the mean of this prior on the null value, 0.05, not 0.5. Yeah.
So what was the prior then? It doesn't matter as long as you know the effective sample size.
And that second value, the mean. So the prior in this case is beta with the first parameter equal to 0.05, second parameter 0.95.
Oh, okay, so it's not symmetric.
Yes, exactly. It's not
Oh,
symmetric.
okay.
Yeah. Other questions? Okay, so I have a question. So Bia, how did you collect those data? What did I ask you to do?
They will be wrong with that.
Yeah, and what were you counting when you rolled the die?
Number ones.
Number of ones. So if you're on Zoom, you didn't see this, but I gave Gao a standard 6-sided die and asked her to roll it 10 times and count the number of ones. So we know that essentially the true value of P should be 1/6. So about 0.17. That's definitely greater than our null of 0.05. So this is a good outcome. This is consistent with what we know to be the truth. Was anyone else instructed to roll a die 10 times and count the number of ones? Michael?
Yep.
Okay, and how many ones did you observe?
2.
2 as well. So one thing that I wanted to do is say, okay, this is the interim analysis. Because suppose that Bao collected the data for the interim analysis, and then we continue the study and Michael is collecting the rest of the data. So what happens when we add in his data? So that was the first prior and 10 observations. Let's go with first prior and 20 observations. So now we have n equals 20 and y sub n equals 4. Let's see, how does this change things? The blue area got bigger. Our probability is now 0.988. It wasn't a huge shift. But actually, if we look at the y-axis, the maximum value on the y-axis is now 5. What was it previously? It was 4. So our posterior is getting taller and more peaked. All right. So that was with one prior. Let's see how things change. I want to know how sensitive our result is to the prior that we chose. So can someone give me a different prior? And we can see if that changes the result that we see. Anyone want to volunteer a new prior?
Let's go with a non-informative.
With a
Non-informative.
non-informative? Okay. So like a uniform?
Yeah.
Okay, great. So we're going to use a sub 1 equals 1 and a sub 2 equals 1 as well. So if we have a All right, so we see the prior has changed. The dashed line is now just a uniform. And we actually increased our probability to 0.997. And so that's because the previous prior was, it was so right skewed. So it had more of its weight all the way near zero. And so it was pulling the posterior closer to zero. Now we have a more evenly distributed prior. And so we have a higher probability under the posterior, greater than 0.2. Any questions?
How important is it to be robust to your prior?
Yeah, go ahead, say more.
Where if you have different priors, the results change slightly, but I mean, not that much. I mean, your conclusion is basically still the same, right?
Yeah.
Does that matter?
That's a good point. I think what you should do is, to better answer this question, the training app does have, so I'm not going to make any changes to the default, even though it's inconsistent with our example we were just using. The training app allows you to conduct simulations. And so what you could do is set the prior to something here, and then run a more comprehensive simulation and see what happens. But yeah, I think that's not a question that I can answer. I think we would need empirical results.
Yeah.
Yeah, probably I can add that comment. So this is the Yuan from MD Anderson. So in general, if you use weakly informed prior, basically it's effective sample size going to measure that. In general, if you keep effective sample size 1, and you can imagine basically what you do is you just add additional 1 patient information into your data. And you can imagine if you applying the sample size, 20, 30, the impact will be very, very minor. And especially in this case, we are not specifically care about a specific posterior exact value of a posterior estimate. We do it's a go, no go decision. And that make a decision even more robust. Maybe the actual value will change from 0.923 to 0.924. But in terms of binary decision, it's even robust, right? If a pass threshold, most time it'll be a pass threshold. So that's one. And the second is, I think a lot of people ask me this question is, can they use informed prior? And I think here is something not totally intuitive when you really use the software or methods. And then when you use informed prior, what's going to happen is the methods and the software will still optimize the decision boundary based on non-informed prior, and then apply the informed prior when you make go, no-go decision. And this kind of anti-intuitive because most people will think I should use the user input prior to determine go, no-go decision. And the reason we don't do that is because remember, in here we control type 1 error. And most time when people input prior information, they less likely put pessimistic prior information. A priori they think this drug won't work. And most time is they test this drug in other indication or subgroup. They believe this is going to be effective. And for example, they put the effective sample size 10 because based on external other information, they think they say equivalent about 10 patient information. They believe this is going to be effective. You put a prior mean at the alternative. For example, in this example, 30%. And when you do that, by default, when you specify that prior, and when you control type 1 error, that's under the none. So in this case, you have the problem, the prior input is conflict with the null hypothesis. And if you still control type 1 error, what's going to happen is you're going to use very strictly go, no-go decision. Basically, you want to compensate the bias due to your informed prior, which against your null hypothesis. And in that case, at the end, you won't have any power gain. I think it's okay, but it's against your initial reason use informed prior. And typically we use informed prior towards the alternative. That means drug effective. So you will end up the results, kind of not what you want. And you still can control type 1 error, but you won't see any power gain.
Yeah.
But the reason you use informative prior is you want to see power gain. So that's why in this software, what we set up is even you enter the informative prior, let's say you put the effective sample size 10, you believe the prior response rate is 30%. Basically, you put the in beta distribution 3 and 7. So alpha equals 3, beta equals 7. That's gonna about the prior mean 30%. And then even you enter that, and what's gonna happen is you still use this default non-informative prior to get go, no-go decision. And then when you do simulation, generate operating characteristic, it will use your informative prior. Because in this case, you will see power gain. That's your intended for. But unfortunately, in that case, when you look at your type of error, it will be inflated. It won't be controlled as you originally entered. Okay, but it's not a failure of the software. It's because this is a conflict. Because if you do that, it's against what you want. So that's a little bit subtle point I want to make here. A lot of people feel it's don't understand what happened behind the scene.
Yeah, thank you so much for sharing that. So that's for people who don't know. So that's Professor Yuan at MD Anderson who created this method. So we do have an expert online with us, which is fantastic. So as you were speaking, I did change the prior effective sample size and recalculate the stopping boundaries, 0, 3, 7, 13. We changed this back to 1. Oh, they changed, okay. The first one changed slightly, 1, 3, 7, 13. But that was really helpful. Thank you.
The reason it changed a little bit because under the sim, when they calculate, they use none as the prior mean. I think when you enter, probably you don't use none, use alternative as the prior mean, right? So here you enter 0.2. But anyway, so if that's the case, if you change a little bit, I can take a look. It should be the same.
Okay. Yeah. Cool. Where were we? Okay, we had done this demo. It seems like people understand this. I was going to do this again. I had asked other people in the room to collect more data, also at DICE. But it's already 10:42. My goodness. So apologies to those of you who did that work. We are going to skip that. Okay, so let's go back to the slides. All right, let's talk about the stopping rule. So big picture, we're going to stop if the treatment is not effective. This means we're going to stop if P is low, but remember we're using Bayesian statistics, so P has a distribution. So we're going to stop if this probability is low. It's the probability that P high is low. So I always find that to be confusing. And this returns us to this question that you were posing earlier. Sorry, what's your name? Jane. Of how low is too low. So we do need some kind of stopping rule, some kind of thresholds. So we're going to stop the trial if this probability is less than this function. So the little n is our interim sample size, the big N is the final sample size, and then lambda and alpha are two other values that we're going to modify, or strictly that we are not going to modify, but the Shiny app will choose for us. So this is a pretty, I don't intuitively have an understanding for what that thing looks like. So I'm going to plot it for us. So I'm just going to choose some values of lambda and alpha. Here we have lambda and alpha both equal to 1/4. And on the x-axis, we have little n over big N ranging from 0 to 1. And then on the y-axis, we have the threshold itself. So you can see this is lambda 0.25, alpha 0.25. I'm going to keep lambda at 0.25, change alpha to 1. We get this line. And I'm going to increase alpha again, this time to 4. And we get this curve. So this is what happens when we change alpha. Going back to our original, 0.25, 0.25. If we hold alpha fixed and change lambda, same shape, but watch the y-axis. That's changing. So alpha is a shape parameter. If alpha is less than 1, the threshold is higher for low values of n over little n over big N. So I just want to, so here we have alpha less than 1. So we've yanked up the threshold for low values of little n. If alpha equals one, the threshold is linear. And if alpha is greater than 1, the threshold is lower for low values of little n over n. And then lambda is going to change the height of the threshold. How do we pick these values? We actually don't. So the Shiny app is going to do this for us. And this is what it's going to do. It's going to find all values of lambda and alpha that satisfy the type 1 error requirement. So we had to set the type 1 error that we wanted. It's going to ensure that we're choosing lambda and alpha that satisfy this requirement. This is actually going to yield multiple values of lambda and alpha. So then the next step is to choose the pair lambda alpha that maximizes the power. So we're ensuring that our type 1 error is what we want it to be. And then we're going to maximize the power. So I'm going to talk about, well, first, before I go into what I found confusing, are there any questions about this? What's that? The little n is the interim sample size. The large n is the final sample size. Not necessarily ideal, because if the drug is truly ineffective, you might want to stop early. N is the largest sample size you're willing to recruit, essentially. So it's the sample size you'd achieve if the drug is actually effective, basically.
Jono, I had a question about the plots you showed us.
Yeah.
So your threshold is your threshold for your probability.
Yes.
So it's greater than 1 in the last one.
Yeah. So if for whatever reason you did have a threshold like this where it rises to the level of 1 almost immediately, this would imply that you would just essentially that you'd have to stop, right? Because we're stopping if the probability is below the threshold. So if the threshold is 1, then we would stop for sure.
Yeah.
Yeah. So I'm not saying this is possible.
Okay.
Yeah. I just wanted to give a bunch of examples.
Okay.
Yeah. But you had a follow-up question? Yeah. No, it's going to be, the small n would be like here for the interims. So like 10 would be n equals 10. We would do our interim analysis, get the number of responses in those 10 values. And then we would do this again for n equals 20. So I'm only using one little n to try and just convey the main idea. But in reality, you will have multiple problems.
Sorry, just to let you know, it's a bit hard to hear. Some people in the audience we can hear, some people we can't. So you may want to repeat the questions.
Thank you. Yeah, I'll do that from now on. So that question was just about what is the little n representing? So interim sample size. So yeah.
When in that threshold formula, there's one little n, like you said, you could have multiple, but at any given interim look, it only depends on the n at that point, right? It doesn't matter with the previous time.
Correct. Yeah. The thresholds is just a formula. The lambda alpha are not going to change. Maximum sample size is not going to change. So it's the interim sample size that is going to change. It's going to give you a value. You'll get your posterior probability and compare it to that value. If it's below, and then you'll end the study. I should say, though, that you don't actually have to do any of this computation. The Shiny app will actually just give you, if I can just show it right now. The Shiny app will give you instructions that are in terms of the number of responses that you see. So we're going to end the trial if the drug is ineffective. So we are going to stop if the number of responses is less than or equal to, some value for a given interim sample size. Yeah. So I'm conveying the lambda and the alpha and stuff, not because you'll need to use it in practice, but so that you understand what's going on. So something that I found confusing, we're stopping the trial if this probability is below some threshold. Under the null hypothesis, we're setting p equal to p naught. So under h naught, won't this probability just be equal to 0? So that was what confused me. What resolved my confusion is that this probability is an integral, and we're integrating out the p. So this probability is not actually a function of that p without a subscript. It's a function of y sub n, the number of responses that we see. So in order to compute the type 1 error, we're going to find the values, or the Shiny app will find the values of y sub n, such that this probability is below this threshold. Then we set p equal to the null value and compute the probability that y sub n does not take one of those values, i.e. the probability that we just continue the trial to the end. And in order to compute the power, we're finding the values of y sub n such that we meet this threshold, or the probability is below the threshold. Then we set p equal to p1 and compute the probability that y sub n takes one of those values. So I don't know if anyone else was confused about that, but it confused me when I first was learning this material, so I wanted to address that. Going back to Zhang's question, aren't there supposed to be multiple looks? Yes, but I was just ignoring the several interim looks to convey the big picture. In reality, the multiple looks make things more complicated. So I thought we could just compute what this threshold would look like for our example. So I'm gonna leave. So we collected data where we had 10 roles and then another 10 roles for a total of 20. Our null hypothesis given to us by Navia was 0.05, alternative 0.3, type 1 error rate. And then we had originally used this default vague prior with effective sample size equals to 1. So I'm going to compute the stopping boundaries. So we would only stop at the interim analysis if we saw 0 responses. I'm trying to get the lambda and alpha. You have to go through all this stuff before getting the lambda and alpha. But once we get this protocol, we can see that its selected values, lambda equals 0.75, alpha equals 0.32. So since that alpha is less than 1, it's going to be easier to stop earlier in the trial. So I thought we could just compute these. Lambda equals 0.75, 2. So at 10 observations, our threshold would be 0.6. So if our posterior probability is below 0.6, we would stop. But again, we don't actually have to do this computation. It's all going to be done for us. So the Shiny app obviates the need for us to worry about lambda and alpha. It's going to produce an easy to read table with 2 columns, number of patients treated, stop if number of responses is less than or equal to this value. Oh. And then that's what we have to use in practice. I'll just go back. So it's going to produce this very easy to read table, and we can just implement that. All right. So that was our in-depth treatment of binary efficacy. So I'm now going to stop and ask, are there any questions? Okay, so this is actually perfect timing. It's 10:54. So we will take a break now. Let's start again at 11 a.m. So I'll see you in about 6 minutes. All right, it's 11, so I'll resume. So the next endpoint is binary toxicity. The concepts for binary toxicity are nearly identical to those for binary efficacy. The difference is that you're stopping now if you see a lot of toxicities instead of few responses. So I think I'm just gonna skip this and move on to the next one, which is simultaneously monitoring efficacy and toxicity. So we have two outcomes. One is binary and good. One is binary and bad. We have two parameters of interest: probability of a response, the probability of a toxicity, P sub B and P sub T. The user-defined inputs are essentially the same as for binary efficacy. They're just more complicated now. So for example, we can have separate interim looks for efficacy and toxicity. So just like we check out the defaults here. Efficacy and toxicity. The defaults give you an extra toxicity interim look at 10 observations. You can change those as you see fit. But there's another way that it's more complicated, so let's talk about it. So with efficacy and toxicity, we have these 4 possible pairings: efficacious and toxic, efficacious and safe, futile and toxic, futile and safe. So this is going to mess with our type 1 error rates. Let's talk about why. We're going to stop the trial if the drug is either futile or toxic. So it's easy to stop the trial in the sense that only one of these two things needs to be true. Either futility or toxicity. We don't need both in order to quit. The alternative hypothesis is that the drug is efficacious and safe. So the null hypothesis has to encompass 3 possibilities: that the drug is futile and toxic. I phrased this poorly. So this should really read that a type 1 error would be, there are essentially 3 ways of making a type 1 error. So the drug could be futile and toxic and we don't stop. The drug could be futile and safe and we don't stop. And the drug could be efficacious and toxic and we don't stop. So instead of null hypothesis, that should really be like type 1 error, essentially. So we have 3 different versions of a type 1 error now. So we have to provide a desired type 1 error rate for each of these. And you can see that here, that there are 3 type 1 error rates for futility and toxicity, safety and futility, and efficacy and toxicity. And so we need to provide these 3. The authors of the BOP2 TE paper, toxicity and efficacy, point out that essentially once you've controlled for 2 of them, the observed type 1 error rate for the third is going to be like way smaller than you need it to be. So by limiting 2 of them to the desired rates, the third one in practice will just be like very low. So don't be surprised if you see that in the simulations. Another way that things get more complicated. So we are concerned with the probability of efficacy and the probability of toxicity. But there's also these other cell probabilities of these intersections. So they will get filled in as soon as we provide one joint probability, the probability of efficacy and toxicity. So that's also something we'll have to provide. We have to provide that under the null and alternative. So we'll have to provide null and alternative hypothesis values for P sub E, P sub T, and then the joint. And you can see that here. There's 3 things we need to input for each of these null and alternative hypotheses. And the authors recommend that you, as you can see right now, the joint probability that's given is the product of the 2 marginal probabilities, implying independence. And the authors recommend that. There's a reason that this is the defaults. The authors recommend that because if you make that assumption, it essentially it's like the least harmful wrong assumption you could make, is the argument. That like even if you're wrong, the negative consequences of being wrong are not that bad. No, I mean, you might have strong reasons to believe that efficacy and toxicity are positively correlated or negatively correlated. I don't know, that's up to you. But this is the default, to assume that they're independent. Yeah, recommendation, assume independence. And you can see these citations for more information. Oh, by the way, so these are links. Obviously, you don't have these slides right now, so you can't click on these links. But what I will do is, after the presentation is over, I will move these slides into the notes repo on GitHub that had been sent out. And then at the end of this presentation, there's also a QR code to that repo, just to make it easy to access that repo. All right, let's talk about the prior. Because we have more than one probability, we're going to use the Dirichlet distribution for our prior. This is an extension of the beta. I'm not going to talk about it in detail. I'm not going to show you the formula. I'm just going to show you some images. So here's a Dirichlet with parameters 1, 1, 1. The Dirichlet, as you can see, it has more parameter values. It can have as many parameters as you want it to have. It's just in terms of visualization, using 3 parameters is the easiest way to visualize this distribution. So that's why I chose 3. So here's a Dirichlet with 3 parameters all equal to 1. So on the x-axis, we have our x1. On the y-axis, we have x2. x3 is essentially determined by x1 and x2. And then you can see from the scale on the right that this is a uniform. It's all at 2. So this is the Dirichlet version of a uniform distribution. Here for Dirichlet 2, 2, 2, where you can see that we're essentially getting this like dome in the center. So this is similar to the beta. When you increase the parameter values, you get a more peaked distribution. If I increase the parameter values even more to 20, 20, 20, it's even more peaked. Once I make it, I can make it asymmetric by varying the values of the parameters, or just one at a time, like so. And then if I make some of those parameter values less than 1, you can see that we get this like super, it's going to be very skewed, similar to the beta. And here's our Dirichlet 0.5, 0.5, 0.5, which you can't even really see, but it has peaks sort of at the very edges. So common priors, uniform prior sets all parameters equal to 1. Jeffrey's prior sets all parameters equal to 1/2. The effective sample size is still the sum of the parameters. And the default in the Shiny app is to match the parameters exactly to the null hypothesis values. So it's an extension of the beta, similar to what we had seen previously. All right, stopping rule. We're going to stop if either of these is true. So if P sub E, the probability of efficacy, essentially if that's low, or if P sub T, essentially if that's high. So I don't know why this is not showing up. The exponent for the second threshold should be a fraction, alpha over 3. I don't know why the fraction bar is not showing up. Guys, can anyone tell me why the exponent in the second inequality is alpha over 3 and not just alpha? Maybe someone other than Professor Yuan?
Oh, because the error is distributed across 3 optimal scenarios.
Not quite. Yeah, we're not yet to the setting type 1 error and stuff. Yeah. it does have to do with multiple possibilities. So the second threshold is for toxicity. Recall that a smaller alpha, as we decrease alpha towards 0, what we're doing is we're pulling up the threshold early in the trial. And we're stopping if our probability is below the threshold. So by pulling up the threshold early in the trial, we're making it easier to stop. So by using this alpha over 3 in the second threshold, we're making it easier to stop the trial early for toxicity. This makes it easier to stop for toxicity earlier in the trial.
Are you saying that's always free?
Yes. I mean, if you use the Shiny app, this is like how it works. Yeah. Yeah.
That almost makes it a judgment.
Say more.
Well, like you want to make it easier to stop toxicity. Is that, it's like, why? Almost.
That's a good question. I can't answer that.
Yeah, I probably I can comment on that. So that's a very good question. Originally, we don't divide by 3. And that's more from a statistical point of view. You can optimize the decision boundary such that you maximize power. The results seems okay from statistical perspective, but when you look at a discrete decision boundary, and you will notice in general it can tolerate higher toxicity. The reason is because if you have very strict early stopping rule, it will affect the power because you have along the way you will make incorrect decisions, stop the trial, leads to lower power. And intuitively, in order to maximize power, and statistically we push the boundary a little bit more relaxed. Now for toxicity, typically, after we have phase 1 trial, and this kind of convention, people kind of want to comply with, right? For example, when you have 2 out of 3 toxicity, 3 out of 6, people in general feel it's kind of not acceptable in practice. So that's why when we tune in this, why we divide by 3, not by 4 or not by 2. And we incorporate this conventional rule in practice. We want that also make it more acceptable for clinician. And when they look at it, they feel it's comfortable. It's not just totally for statistical purpose. We control type and error and maximize power. We also want it kind of consistent with a lot of practice.
I have a question. Why adjust that parameter as opposed to the null toxicity rate?
Yeah. So for non-toxicity rate, again, I think for statistician, it's easy to understand because those two are 2 sides of the equation. But for clinician all time, they have kind of convention, right? Like more than 33%, it's not acceptable, 20 to 30, that's okay. So we want people feel more comfortable with the setup, the non and alternative, and we tweak the decision boundary.
Okay. So for when you keep the null in the 20 to 33% range, which is what people are kind of comfortable with, the alpha is the parameter that needs to be adjusted so that you get the 2 out of 3 or 3 out of 6.
Yeah,
okay.
Exactly.
Yeah.
And in principle, statistically, in principle, we don't even have to have the same alpha. And you can have alpha 1 for efficacy, alpha 2 for toxicity. And in that case, leave you total freedom to specify those. But if you do that, and then you also have to specify another constraint, for example, under what toxicity I want to stop. And we feel it's just too burdensome for user. That's why eventually we take this kind of middle ground approach, use same alpha, but we empirically choose divided by 3 to kind of balance simplicity and a practical consideration.
Thank you. Follow-up questions?
I have a naive question. If you assume the toxicity and efficacy are independent, that seems to be a recommendation, and then And then follow up just what Professor had mentioned, that we could even choose separate outputs for the toxicity. Why do we need to do this combined framework? Because we could have used the efficacy framework, single endpoint, and the toxicity framework, and it would still work out. So we had different rules and, for toxicity for efficacy. And in fact, we don't even need to use the same number of patients for the interim looks if we do it independently.
Yeah, that's a very, very good question. And what you just said, it's a lot of time people used in practice. And the main advantage is in that approach, it's pretty simple. But statistically rigorous speaking, the operating characteristic you generated based on that rule, it's not accurate. So because in that time, when you generate it, what's your stopping rate for futility, what's your stopping rate for toxicity, you assume then the other side doesn't happen, right? You can imagine in the case it's futile and also toxic, the stopping rate will be higher than when you look at either table, even based on either based on toxicity or efficacy, because there's more chance stop, because those two rules are gonna interact with each other. And when you consider the jointly, statistically it's more rigorous because the operating characteristic gonna reflect what really is the true operating characteristic. And then your question regarding of why, when you design the trial, use independence, does that, equivalent to the case you just use two marginal stopping rule? Actually, they are different. And when we assume independence, it's just used to optimize the power. And when you really implement the design and also get that stopping rule, it's considered a correlation. And that's why when you look at the operating characteristic and go to the simulation part, that tab, it will ask you to specify the correlation. And it depends on what correlation you have, you will get a different operating characteristic. So that's what you should anticipate in your real trial. And the reason we do independent for design the trial is because in many cases, it's so hard for investigator to specify this correlation. And again, we're trying to balance, simplicity with how rigorous it is. And in the paper, we showed the stopping rule, the type 1 error, is the monotonic function of this correlation. And so that means if they are positively correlated, and when you assume independent, you're always more conservative. And when they are negative correlated, and we showed it's not sensitive at all. So that's why we feel when you use independent and for user, you don't have to worry too much about when you set up the trial. The boundary, it's very robust. And once you get the boundary, when you evaluate the true operating characteristic, so in that stage, you will consider correlation. And in that case, it's simple because you can enter different correlations, see how operating characteristics change. But remember, in this page, this is the design phase. You have to enter one value, get your boundary. It's like a power calculation. You have to give a certain value assumption so you can calculate the sample size and the power. And the effect sample size may be different from what you assumed. And that's very similar in that situation.
Thank you. Do you have a follow-up question, Susanna? No, okay. All right. Okay, next option, multiple efficacy. So for this endpoint, we can have two outcomes, both are binary and good, two parameters of interest, probability of a response for the first outcome and the probability of a response for the second outcome. So at first blush, it seems similar to what we just talked about, except instead of a toxicity, we have just a second efficacy outcome. For user-defined inputs, there's just one set of interim sample sizes for both measures of efficacy. For the desired type 1 error rate, there's only one that we have to consider. And I can talk about why that is. So here are the possible outcomes, both efficacious, efficacious for one and not for the other, or both futile. We're only going to stop the trial if we decide the drug is futile for both endpoints. So this is a stricter stopping criterion in the sense that like both endpoints, the drug would need to be futile in both cases for us to stop. And so a type 1 error would be made if the drug is futile for both endpoints and we don't stop the trial. And there's only one way for that to happen. So you can see that if we have multiple efficacy, there's only one type 1 error rate that we have to select. It's not allowing me to click on what I want to click on. Sorry. Here we go. Okay. In contrast, so there are 3 ways we could make a type 2 error. The drug is efficacious for both endpoints and we stop the trial. Drug is efficacious for endpoint 1, we stop the trial. Drug is efficacious for endpoint 2 and we stop the trial. Oops. So 3 ways we could make a type 2 error. With BOP2, recall that we're first controlling the type 1 error rate and then maximizing power. Maximizing power is the same as minimizing the type 2 error rate. With 2 binary efficacy endpoints, we don't minimize multiple type 2 error rates. I know that there are 3 different like versions that I just went through. We're just minimizing the rate of stopping the trial even when the drug is efficacious for both endpoints. So we're just minimizing like that version of the type 1 error. I went through that pretty quickly. I apologize if that was confusing. It's actually, this is simpler than what we were just talking about. So hopefully that's reassuring. All right, more user-defined inputs. We do have to provide P1, P2, and the probability of both responses at once. So we, again, are having to provide this probability of the intersection, or the joint probability. And then we have to provide a prior within certain constraints, those constraints being, we're using the Dirichlet. Like I said, we're stopping the trial if both of these are true. So, you know, it's just 2 versions of the same thing, essentially. Note that now we're using the same lambda for both endpoints and the same alpha for both endpoints. So we're not dividing anything by 3 this time. All right, I know that was pretty quick, but that's multiple efficacy for you.
Yeah, can I make a quick comment on this?
Yes, please do.
I think it's more about terminology. And in the software, we use a multiple endpoint. That's in order to, that's the terminology FDA use in their guidance. But in medical field, a lot of time, we also call it a co-primary endpoint. I think that's one point that I want to highlight. So essentially, this is the case when you stop only when both endpoints failed. So that's exactly what it means. But in terms of terminology, I see, you know, some medical journal, some try to call it co-primary. But the reason we use multiple endpoint is because to be aligned with FDA guidance. So in FDA guidance, multiple endpoint means you any point is positive, you see the trial is positive. And co-primary endpoint, according to FDA guidance, it's in the case you need both endpoint positive to be claimed your trial is positive.
Sorry, I had to step away for a second, but just to confirm, so in this case, if you have a positive on either endpoint, the overall is positive.
Yes, that's right.
Okay. So it's something like objective response rate and like clinical benefit rate.
Exactly.
Or like PFS6 or something like that would be like your 2 binary endpoints in the oncology setting.
Yeah, but a lot of time people in publication, a lot of people also call it co-primary. That's why here I don't want people to get confused.
The original extension of the Simon's 2-stage, what is it? SIL and something called it co-primary, I think.
Yeah, even a lot of New England Journal of Medicine paper right now, they also call it co-primary. But I think the reason we choose here is because FDA have guidance on that
they
scenario.
don't like the term.
Yeah, they call co-primary only when the case you need both endpoint positive to claim the trial is positive.
Okay.
In this case, the multiple endpoints, they both have to be observable at the same time, right? Because if you,
Yeah, that's true. And in practice, a lot of time you can pause a little bit, wait both mature before you can make a decision.
Yeah, that's how we sometimes do it on the GYN side. Like if you don't hit your response rate, then you wait to see if you get enough progression free at 6 months. And then if you see signal on that, then you continue. But if you don't even see it on that, then that's where you stop for futility.
Yeah. And if you see one during the interim, if you see response rate it's past the threshold, then you don't have to wait. You just directly move forward. So in practice, it's not that logistic challenge as it seems to be.
Okay, ordinal efficacy. So maybe I just want to point out, I'm going to show ordinal efficacy. So you can see that we have probability of CR, complete remission, and probability of CR or PR, so complete remission or partial remission. So right off the bat, the terminology is coming from cancer research, unsurprisingly. Obviously, you could use this with other outcomes that are not specifically the scale of complete remission, partial remission, stable disease, progressive disease, etc. But that's just the notation that appears in the Shiny app. So the best level, if we're thinking of just an ordinal scale with at least 3 levels, the best level is CR, the second best level is PR. And then maybe I'm telling tales out of school, but at least statistically, it seems like the other levels are essentially irrelevant. And I can make a case for that later. But we're focusing essentially on 2 parameters of interest. So here you can see that we have the probability of CR, our best option, complete remission, or the probability of either CR or PR. For me, and just the way to get my head straight about this, I'm thinking of probability of CR and probability of PR, just separating them. And then the joint, or not the joint probability, but the probability of the union is going to be just the sum of these because they're mutually exclusive. I apologize if that's confusing, but it made more sense to me. So we're going to stop the trial for futility if both of the following criteria are met. So let's focus on this first one. So essentially, if the probability of complete remission is low, or the way it's written here, if the probability that it's high is less than some thresholds. So low probability of complete remission, that's considering, we will stop if that's true, and also if this following thing is true. If the probability of either CR or PR is low. So there is a lot of notation on this slide. I apologize. We're stopping for futility if both the probability of complete remission is low and if the probability of either complete remission or partial remission is low. So I admit this is a lot of notation. In order to make this make sense to me, I plotted it. So I want to show you my plots. Oh, and I do just at this point that it's the same lambda and alpha. So at the risk of oversimplification, let's just say we're going to stop if the probability of complete remission is under 0.15 rather, and the probability of either complete remission or partial remission is under 0.3. And I got these values, they're just the default values from the Shiny app. So stopping would correspond to the region in the plot with both horizontal and vertical lines. So here's the plot. It's smaller than I want it to be, but I can use my mouse to highlight different regions. Okay, so here on the vertical lines, so the area that's shaded with vertical lines, that's where the probability of complete remission is low, is below 0.15. My mouse disappeared. Oh, you can see it. It should be set up so that you can see it by point. So P sub CR is on the x-axis. So if this is low, that's the vertically shaded region. The horizontally shaded region you can see is this triangle. That's where the sum of both PCR and PPR is low. So the probability of either complete remission or partial remission. We're going to stop the trial if both of those things are small. So that's this double shaded region. This like, what is that shape? Rhombus? I don't know. Anyway, this shape, if we're in here, we're going to stop the trial. I want to show a contrasting image. So I want to compare this to the situation where we would stop if, like, the probability of CR were low and the probability of PR were low. So that would look like this. The overlapping region would just be this rectangle. So this is not what we're doing. We are doing this, we're stopping if we're in the double shaded region. You can see that here, the double shaded region, it's smaller. It doesn't have the corner that this one does. So my interpretation of this is that it's slightly harder to stop because we're stopping in like a smaller region of this outcome space than here. But I think we need more context than that. So y'all are the experts. Can you tell me about, so is this a common thing that you're doing where you're stopping a phase 2 trial if both of these things are true? Where you have these two things to consider, both this like probability of complete remission and then this probability of either complete remission or partial remission?
Yeah, I can provide a little bit
Yeah.
clinical context for this specific why we developed this. So I think if you're familiar with the cancer trial, most time we use objective response rate in a phase 2 trial. That's CR plus or PR. And in some trial, we noted that, and especially for some target agent, so the ORR is not that as high as we want. But when we look at the most response, they are complete response. So in this case, the PI come back to us and say, oh, yes, it doesn't reach our benchmark, for example, 30%, but it's only like 20%. But those 20% are all CR. That's highly desirable. And they don't want to stop the trial. So that's why in here, we put the more desirable CR, put another level. So in case that happen, it give another chance to let the trial move forward. So that's the clinical rationale behind it. I think it quite depends on the setting and the drug, and sometimes it's reasonable. Sometimes it may not necessary. You just based on RR, it's not good enough. You're going to stop the trial.
Yeah.
In multiple myeloma, the endpoint is MRD CR. There are people that don't get MRD negativity that are in CR. And that composition is definitely of interest.
Okay. Could you hear that online? So that was not another question, but an example from a specific disease type where it would make sense to apply this.
Yeah.
We also have the immunotherapy where we're looking at the response rate, but then turns out the other thing, sometimes again, looks like your, the response rate may be low overall, but there are these sustained stable diseases which is high, if you combine, so the clinical benefit rate is high, but the response rate itself is not, it's low. So you don't want to stop the trial.
Yeah. So right now we are working on a extension of this to consider both ORR and DOR. As you said, sometimes we don't see enough response, but those response are durable. We are working on that setting. We see sometimes it happen in practice.
But Bial, the setting you're describing, we use the previous design for those, the multiple efficacy one. So you have like response rate and then either clinical benefit rate or PFS6 as your second one. So those take into account like basically they don't get a response, but maybe you have prolonged stable disease, and then that would count. So that it is possible with the previous design to do that kind of study.
That's right.
Right, but stable disease is also one of the ordinal responses in RECIST. So you could also look at it this way.
I suppose, I mean, the PR and the CR in this context are like just labels, right? You could consider like stable disease duration greater than 6 months as your second one, like, right? You could make CR be like just response, any response, CR or PR. And then you could make the PR one be category instead be stable disease greater than 6 months.
You can.
Yeah, exactly. So you can look at one of the CBR, clinical benefit rate, the other is ORR, right? So the clinical benefit can include a stable disease.
Yeah,
Yeah,
I mean,
absent a duration. He just said that that's gonna be the next step, but not here. If you're gonna define it by a certain duration, as Nabila just said, I don't get that. It's not the same scale that you're looking at.
are you talking about
Like an overall, like she said, like, you know, the levels of resist.
versus a time to event?
Or she said you have to require the 6 months to establish
Right, I mean,
CBR.
Those are different options. I don't
I
think.
don't feel like that's the same situation here.
I think it's just like the question is whether you would in this case, I mean, I think it's much simpler in the multiple efficacy one. In this case, I think your question is basically if you want to turn it into an ordinal. I mean, we actually put these in the paper in that order, right? We put CR first, then PR, then for stable disease, we put stable disease greater than 6 months because I mean, of our setting of like I'm thinking the most common way we put this is GYN cancers. So stable disease, we use a threshold of 6 months in the GYN cancers. So stable disease greater than 6 months is kind of the next best response that somebody could get. And then stable disease less than 6 months is kind of like, it's okay, but it's not very durable. And then PD is like your worst category. So we use the kind of ordinal, creating an ordinal out of the best overall response thresholds. And so I think that you could, I mean, I haven't gone into the mathematics of the method, so, but it seems to me like your CR and PR can be defined in any ways you want. It's whatever ordinal category. It just has to be the higher category than the lowers.
Yeah, maybe addressing the terminology. So it's called ordinal efficacy here. I believe that in the original BOP2 paper, it's referred to as nested efficacy. So the other key idea is that you have these categorical outcomes and you have one of them here denoted CR. And then you have this other thing which incorporates both your original outcome and something else. So it's nested within something else.
And so that's, yeah. So then objective response and clinical benefit are nested. If you have those two endpoints, the way they most commonly defined are nested. Objective response are usually defined as CR or PR, and then clinical benefit is CR, PR, or stable disease, sometimes with a duration, sometimes without.
And the reason in here we eventually decide not use nested because actually you're right. So here intended to be nested. That's more for a statistician. But eventually we decide to use ordinal because a lot of clinicians don't understand what do you mean nested. That's why here we put the ordinal efficacy. But Jonathan is exactly right. So in here it's intended for the case, it's nested. And the reason we distinguish multiple efficacy and ordinal efficacy, mathematics a lot of times they have a lot of commonality, and you can use either way. If you understand what happened, you can take both, achieve the same goal. But here, the reason we separate it, it's more from a clinical perspective. And also, and because it's nested, that's why in terms of modeling, how you generate the data is slightly different. Because remember, when you have multiple efficacy endpoints, you have to specify the correlation. Because in that case, we consider those two, kind of conceptual separate endpoints, they may correlate. But in here, you will notice we don't ask you end the correlation because we assume they are nested. You don't have to specify the correlation because it's one endpoint. It just have different cutoff for the level.
Awesome. Thanks, everyone. So we just have one more endpoint to consider, which is time to event. I'm very curious for your perspective on this. So our outcome is a time to event. I have some notation to introduce. We're going to consider Z sub I to be the time to event for the ith participant, U sub I as the censoring time, and we're going to observe the minimum of those two things, which I'll denote with T sub I. And the parameter of interest is mu, the median survival time. Note that it's the median, not the mean. Okay. So we have to select all the usual stuff. We have two additional settings, accrual rate and follow-up time after the last patient enrolled. So the Shiny app assumes there's only one reason for censoring, which is that anyone who hasn't had an event by the end of follow-up time for the last patient enrolled gets censored. So as far as the Shiny app goes, it's not gonna, there's no way to program into it like, oh, assume these people are lost to follow-up or whatever. There's just this one version of administrative censoring, which depends on your accrual rates and your follow-up time. I don't know, have you have thoughts on that? I'm curious if you do.
Yeah, so ideally we should incorporate censoring process. But in this setting, we assume administrative censoring at the end of the trial. I think potentially we can add that, but in general, I think the impact on the operating characteristic and also decision boundary will be pretty minor. Because at the end of the day, when you evaluate the time to event, it's mainly driven by the events, not by the censoring, right?
Yeah,
but because we use the exponential model rather than log rank. If you use a log rank test, it doesn't matter the censoring. It's more number of events going to decide the log rank test statistic. And then here, because we use underlying what happened is we assume it's a time to event follow exponential distribution. And it sounds very restrictive. The reason we do that for two, one is in that exponential model, we have closed form. That's why you can do a real-time calculation. And also it allows us to give a boundary a priori. You can pre-tabulate it. You don't have to real-time calculation. You just sum up total event follow-up time and can look up table, make a decision. But of course, I think the alternative you can use a Weibull model, more or piecewise exponential, more flexible. Methodology-wise, actually, we develop methods for that. But in that case, you won't have any pre-tabulated boundary. You have to do real-time calculation.
Got it.
And for the go no go decision, in general, even exponential is misspecified. It's quite robust, as we showed in the publication. So that's the reason we choose this approach.
Thank you. In terms of the prior, we assume the median has an inverse gamma prior. The Shiny app constrains the values of A and B. So N naught is called the effective sample size. I don't know why, again, the second parameter should be a fraction. I don't know why the bar is not displaying. And then mu naught is the median survival time under the null. Oh, and then as Professor Yuan was just talking about, the likelihood is that we're assuming that the time to event is exponentially distributed. Again, mu is the median. And then you'd already addressed that. So for the effective sample size, if mu is inverse gamma with parameters, hyperparameters A and B, then the posterior distribution is this thing where we have this notation. So the first parameter increased by the number of observed events. So it seems to me like the first parameter should be the effective sample size, not the effective sample size minus 1. But that's a minor point. Regardless of naming, we can look at various possible priors. So here we have a prior where the null median is equal to 6 and the effective sample size is equal to 0.05. This is the default. So I have mu, the x-axis going from 0 to 2. And then you can see what this looks like. So what I'm gonna do, keep effective sample size at 0.05 and increase the median to 10. So it's shifting the weight of the distribution to the right. That makes sense. We're moving the median to the right. And the y-axis is getting lower. So we're flattening out the distribution by increasing the median. Okay, so now what happens if I decrease the effective sample size? It's getting more peaked. You see the y-axis has increased. So by decreasing the effective sample size, we're actually making this distribution, the prior, have less variability. It's becoming more peaked. And by increasing the effective sample size to 0.5, we're flattening it out, which seems contrary to what I would want. I would think I would want a lower effective sample size to correspond to less certainty. But I don't know. We're running out of time. I know we have only 13 minutes left. So I just we could look at the relationship between the prior and the posterior. I had a whole thing. This might take too long, especially since, Suga, how many trials did you actually end up being able to do? How many times did you roll the die?
6.
6 times. Yeah. So I think I had some real experimental data we could use, but I think we're close to the end, and so maybe we're just going to skip the demo. So in terms of the stopping rule, this should look familiar. M sub n is the number of events that we see. TOT stands for total observation time. That's the sum of all the T's. We're stopping if, the probability that our median is really big is small. And it's worth looking at what the Shiny app produces. So I'm just going to show you, I'm just going to use the defaults, 2 interims, 15 and 30. You can see these accrual rate 1.5, follow-up time of 12. Median survival time in the null is 6. The alternative, we have 10. Type 1 error rate, 0.1. Let's look at what the stopping boundaries are. So the Shiny app, as usual, makes things very easy for us. It's going to say, okay, for the interim, we have treated 15 patients. How many events do we have? Let's say we have 6 events. We're going to stop the trial if the total observation time is less than or equal to 55.45. Obviously, there are no units here, so you're going to have to know what units you're talking about in your actual trial. But this is how your decision-making process will go. You'll look at the number of events that you actually observed, and then stop the trial if the total observation time is less than or equal to some value. Is anyone, some brave volunteer, maybe not Professor Yuan, want to talk through the reasoning for why we're doing this? Why is it that for a fixed number of events, we're going to stop the trial if the total observation time is less than or equal to some value? So when we get to our interim look at 15 patients, we're going to say, how many events did we observe? I don't know, maybe we'll say 6. And then we can go over in that row and say, we're going to stop if the total observation time is less than or equal to 55.45. Whether that's months or weeks, I don't know. But so why is it, sort of make the argument to me, why does it make sense to stop if the total observation time is less than or equal to some value for a given number of events?
You could think of the events over the total observation time as kind of a crude hazard calculation. And so basically, if the smaller your observation time, the bigger your hazard for the fixed number of events. And so your hazard is too big, basically.
Great. Yeah, thank you. Love that. So we remember we're stopping for futility. So we're stopping if the drug doesn't work. That means we're going to stop if we have lots of events like really soon. If the drug worked, we'd have to wait a long time to see events. So we want to stop if we have a bunch of events right quick. So if we have 6 events in a very short period of time, our drug doesn't work. So we're stopping our trial.
If we don't get the accrual rate right, then how does that play into this accrued observation time?
Yeah, that's a good question. Maybe Professor Yuan, do you have
Because
response?
we hardly ever get the accrual rate right in practice.
Yeah, that's fair.
Yeah, I think the core rate in here, it doesn't matter too much. It's more impact operating characteristic. Because in the calculation, exponential inverse gamma model, I think if a statistician imagine that, and that calculation doesn't involve accrual rate at all. The accrual matters is because when you do simulation, you have to simulate the patient enroll process, and in that time, by the time you observe, for example, 6 event, and that's the accrual rate going to impact what's the expected total observation time, and that's why accrual rate needed for that purpose.
Can I clarify how this would be implemented? So you have to treat your required number of patients in this case, right? You have to treat 15 patients and then wait and observe them and see how these events, it's not like a binary endpoint where sometimes you've treated, let's say your threshold is 2 out of 12, but you treat the first 5 patients and you get 2, you've already hit 2 and then you can continue. Or like if it's the opposite for toxicity, you've hit your boundary already, it's too toxic. Even if you haven't got the full enrollment of the interim, you can make a decision because you've already hit your threshold that you know. But in this case, you have to enroll all your patients and then you can assess them.
Yeah, so you reach the interim time pre-planned. So for example, you want to enroll 15 patients, and you finish when you complete the enrollment 15 patients, you look at the number of events you have, and then you look at the total observation time. And the number of events is a random variable, right? Depends on at that point you want to make a decision how many events you have, and you look at different row to make a decision.
So you can make a decision straight after you enroll that 15th patient if you want?
Yeah, it's kind of you can make it in real time.
Okay.
But the difference is when you have more events, wait longer time. In that case, your decision is more reliable, right? And for example, you make that decision immediately, you will anticipate less event. And although in here we can make a decision, but the decision, how reliable it is, it depends on how number of events you have.
Can I ask a quick question? This is Noah.
Yeah, go ahead.
So and maybe this is a dumb question, but
How's
can we talk a little bit about just that first row? So 0 events. I think if I'm understanding correctly, the row below, you're saying if you've seen 1 event by a little over 1 year, when total observation time is a little over 1 year, you're saying that's too many events for that time period, so you would stop. For 0 events, that same time is a little over 5 months. But you would still stop even though you haven't seen any events. Is is that because maybe that would be like an indicator that patients aren't staying in follow-up for long enough and maybe dropping out, and that's kind of where that's coming from? Or it just seems a little counterintuitive that you would stop when you haven't seen any events.
Yeah, that's a good point. I agree. I think probably we should remove that role. And the reason we have that is just pure based on a mathematical calculation.
Right.
And in that case, the likelihood, the probability, it's gonna exceed the threshold we want to stop. But you know, from practical point of view, I agree. It doesn't make too much sense because the reason we can make that decision in this case is we totally rely on the exponential distribution assumption. If the assumption is absolute true, and when you make the decision on average statistically, that's the right decision. But I agree with you. In practice, first the exponential distribution may be wrong, and you don't have any events, you stop it. It seems doesn't make sense.
Okay. Yeah, thank you.
I do have a follow-up question that I hope can be answered quickly. So posterior, if I go back to it, it's a function of the number of events that you observe and the total observation time, which those are going to be a function of the censoring. So even if you're making this assumption that the time to event is exponential, if you incorporate the censoring from, you know, administratively censoring people after the follow-up time is completed, you just get like a truncated exponential, which shouldn't be that much. I would imagine that's not that much more difficult mathematically to, like, it's not that much less tractable. So, but you had said, Professor Yuan, that when it comes to computing the stopping boundaries, you're not incorporating the censoring at all. Is that true?
So yes, so if it's a follow exponentially, this parametric model is correct. When you look at this posterior, so it doesn't matter. You know, it's only two quantity enter this equation. One is number of events, right? That's delta I. The other is total observation time, Ti. So in here, it doesn't matter how many, you know, it's only dependent on those summary statistics. And so even when you have censoring, what's going to happen is you're going to reduce number of events. But given you have the same number of events, what matters is Ti. And the equation won't change at all because this posterior is going to be exactly the same. And when you set it the same cutoff, it's going to exactly the same decision boundary. The only thing is, if there is a censoring, given the same sample size, the same follow-up time, you won't get the same number of delta. So I think I don't think the decision boundary is going to change even you have a censoring process. It will only impact operating characteristic. So that means given that stopping boundary, you are unlikely to reach a stopping boundary because the delta I going to different, the Ti going to different when you have censoring versus you have no censoring process.
But you're computing the stopping boundaries to match a certain type 1 error rate, and then you're maximizing the power. So those are probabilities that you're computing, and those probabilities are functions of these random variables, for example, the total observation time. So in order to compute the type 1 error rate, you need to compute the probability that you observe a specific total observation time under the assumption that the median survival time is equal to some value. And so you need to know the distribution of the total observation time. And that's going to be a function of the censoring. I think we got through everything, and it's exactly new. So the first thing I want to do is say thank you so much for coming, Professor Yuan. I really appreciate having you here and answering all these questions that people had. And then the second thing I'll say is if you want to see these slides or any of the notes, they are in this repo. You can use this QR code or go to this link. And I'll put the slides in that repo after sometime later today, probably. And then, yeah, I think that we can stop there. Yes, that's it, unless anyone has a final follow-up question they want to get in. Okay, all right. So thanks again to all of you for coming, and thank you, Professor Yuan, for coming and answering so many questions.
Yeah, thank you. Excellent presentation.
Thank you so much.
Thanks, Jono. Bye.